Let's move on in the digestive system to talk about the actual digestion and then absorption of the nutrients. And as we talk, think about what we've already discussed, how the chyme, the bolus and the chyme is moving through the digestive tract through contractions of the muscle. So as we've already seen, we have a lot of control over the muscular movement and the progression of the nutrients through the system. And this is no different when we're talking about the secretions that are actually containing mucus or enzymes or bicarbonate to aid in digestion. And so this comes through the afferent neurons in the same way that the other types of control is done, through afferent neurons responding to chemical cues or touch receptors or stretch receptors. Especially distension is an important part of this. And it is a reflex. Usually these secretions will be automatically triggered by the stretching or the irritation in the digestive tract. And it's also controlled, as we've already looked at, through the hormones. With most of the secretions, we're going to get an increase with parasympathetic and a decrease with sympathetic. As the arrow shows, there is some exception to that with the sympathetic stimulation. Sometimes we'll get increased secretion or changes to the secretion associated with the sympathetic system or sympathetic stimulation. The best example of that would be with saliva. You get increased salivary production with the parasympathetic system in terms of volume and content. You get a change in the secretion with the sympathetic. So it's mostly decrease in saliva production, but it's less aqueous, more mucus type secretions when the sympathetic system is activated. Okay, so talking of salivary glands, let's look at how the glands are innervated. So as I mentioned, you have the parasympathetic regulation that is feeding back from taste stimuli through the glossopharyngeal facial nerve that synapses when there's a taste on the tongue that will trigger salivation within the various salivary glands shown here. Salivation can also be triggered by seeing food and smelling food, but this is the best direct connection that we can make between the taste of food. And you do need saliva in order to be able to taste the food, so it's kind of like a feedback loop as well. All right, so let's talk about what saliva is. It helps to form the bolus, which we discussed already. It helps to begin digestion in terms of mechanical digestion because it solubilizes the food, and it also initiates starch digestion because it contains amylase. The other thing that saliva is important for is providing a barrier in terms of defense, in terms of the immune system. And so we get production of IgA and lysozyme, which helps to break down bacteria by the salivary glands. And when we have decreased saliva when we're sleeping, that can actually allow for the buildup of bacteria in the mouth. So one good reason to brush your teeth after you wake up in the morning. So we're going to shift right into gastric secretion and talk about what's happening in the stomach. So we have various cells in the stomach that are important for both protecting and producing the enzymes that are working there. And the reason that we have the high content of hydrogen ions, or the low pH, high acidity, is because of the parietal cells and this mechanism by which pepsin, which is the enzyme that breaks down proteins, functions. So the other thing that's shown on this slide that it's not highlighted are the glands up towards the top, the blue ones. Those are mucus-producing glands. So in addition to having the production of the pepsin and the HCl, we also have mucus to protect the lining of the stomach from the high acidity. All right, so this is fairly straightforward. It's from an A&P textbook, but it shows how the chief cells in the glands of the stomach, specifically the gastric glands, secrete pepsinogen. And this is what's known as a zymogen. Zymogen is an enzyme that hasn't been activated. And so the parietal cells need to secrete the HCl to allow for that acidity to cause the activation of the peptide, of the pepsin. And then the pepsin itself, once activated, will break down proteins. Now, this mechanism helps to protect the stomach from the pepsin because you don't want the cells or the lining to be degraded by the pepsinogen. So what you really want is to require both the chief cells and the parietal cells to secrete, and then have those secretions come together in the lumen of the stomach, so that activated enzymes are mostly present in the lumen, and that helps again to protect the lining of the stomach, the mucosa of the stomach. We see this type of mechanism throughout the digestive tract. We see it in addition especially in the duodenum, where many of those enzymes have to be activated, and that helps protect the pancreas, which produces the enzymes, from the enzymes themselves. Because remember that the cells that are secreting these things are made up of proteins and lipids and carbohydrates. And so if you have active enzymes being secreted directly from a cell, that has the potential to damage the cell or the surrounding cells. So again, I show this as one example of a mechanism that is taken advantage of by in many of these parts of the digestive tract to allow for an inactive or zymogen to be secreted, and then it becomes activated through, in this example, it's through HCl. In the other examples, it's various mechanisms. Okay, so how HCl is secreted is really interesting because, remember, think about it. HCl is pretty caustic. It can't just be produced in a cell and secreted, or it's going to cause degradation of that cell. And so what actually happens is you get secretion within the lumen of what's called canaliculi. They're little tiny ducts in the cells. Your book has a diagram of them. So you get secretion right towards the outside part of the parietal cells of hydrogen and chloride. And then it comes together to form HCl. And again, this is to help protect the cells from that caustic HCl. So I realize that this diagram probably looks intimidating, but take a minute and check it out. The top part there in red, where you have the hydrogen ions coming out, that should look familiar to you. We've got a chloride shift going on, moving bicarbonate in and out of the cell. We have CO2 moving back and forth. That is similarly used in the respiratory tract. Then below that, we have the sodium-potassium ATPase pump. That is used in neurons to exchange sodium and potassium. And what this is doing is it's creating a gradient that allows for the movement of so the input of the potassium is creating this gradient. It allows for the movement of hydrogen ions out of the cell. And then the sodium ions being moved across the cell to again enhance to allow for the function of the ATPase pump is allowing for that movement of the chloride out of the cell. And then we've got osmosis through water movement. So initially this might look intimidating, but it's actually really interesting, and you've seen these transporters before, so they should be familiar to you. Notice the pH 2 can get pretty low, so you can get a pH of all the way down to 0.8. Usually the stomach is referred to as having a pH of 2 because once this HCl solution leaves the proximity of the parietal cells and mixes up in the stomach, it's going to come up a bit in terms of pH. Okay, so we see this, the important point here is that the osmolarity of the secretions is always neutral, or isotonic, I should say. So we've also always got an isotonic environment here. And so we see changes at low secretory rate versus high secretory rate in the ions that are being exchanged to keep this isotonic nature. So we either have high sodium chloride or high HCl depending on whether secretion is occurring, and that again is to keep it isotonic, to keep the cells from becoming hyper or hypotonic. All right, so I mentioned a mucosal barrier earlier. The stomach does have a thin lining of mucus and has a tight junctions and of course the cell membranes that help keep the HCl in the lumen, not entering back in, keeps the hydrogen ions not entering back into the blood or the circulation or the mucosa of the stomach. And as the HCl is being created, extra hydrogen ions are transported back into the lumen to keep the cells at a functional pH. All right, so let's look at the pancreas. It is creating a majority of the enzymes to break down the main nutrients. Those main nutrients being proteins, fats, and carbohydrates or starch. The key here is that the pancreas empties into the duodenum right at the point where the food goes from the stomach into the duodenum. This allows for the enzymes to come into contact with that chyme that has just been well broken down in the stomach, but the acid has to be neutralized or it will damage the enzymes and it will damage the mucosa of the duodenum. And so in addition to the enzymes coming in from the pancreas, we also get bicarbonate coming in and neutralizing those enzymes. So the other thing that is secreted is secretin, which allows for bicarbonate to be secreted and increases production of that within the pancreas. And as we mentioned earlier, it's nature's antacid. And so the mechanism by which this occurs is very similar to what's happening in the, or similar to what's happening in the respiratory tract. So we go from HCl and we end up with CO2 and water and sodium chloride. All right. So bicarb, it has a similar secretion mechanism as the HCl in terms of its coordination between different transporters. So we need to take bicarb out of the blood or out of the interstitial fluid, transport it across the cell of the pancreas and into the lumen. That lumen is going to end up in the duct, which will empty into the duodenum. And so this gradient is set up such that we can allow for this transport, again, through the chloride shift to bring the bicarbonate out and into the lumen of the cell. And that is enhanced by the exchanges occurred here in the sodium-potassium pump and the hydrogen ions being exchanged. And it's all to create this environment through which we can either bring in HCO3 bicarbonate or make it, as you can see there, and therefore produce it so that it can be secreted into the lumen. And there are specialized cells that do this, the pancreatic duct cells, which you can actually see on histology. All right. So we've mentioned this already, but the pancreas is under a lot of regulation. So there's this feedback from secretin, which allows for response to acid and therefore increased production of bicarbonate. We also have feedback from CCK, which will increase production. So both of those are going to be secreted in response to acids or stretching and things like that. And that will cause increased production and secretion by the pancreas. Notice we also get vagal stimulation to help with the enzymes, and so we do get feedback from the parasympathetic system as well. All right, so let's talk about the liver. We're not going to get into too much detail. All I really want to talk about is the bile and the bile acids that are involved in the liver. So we have a need to break down lipids into smaller units, into droplets, so that the enzyme, the lipase, can have access to them. And so this is done through the bile salts, which are found in bile. Bile is produced by the liver. It's specifically produced by the hepatocytes of the liver, and it is stored in the gallbladder. Now, the liver, as we talked about earlier, responds to CCK in the same way, and the gallbladder responds to CCK in the same way as the pancreas. So when you get an input of fat into the duodenum, that's going to cause increased secretion of bile. And then that bile, remember, fat, lipid is hydrophobic. So it's going to tend to form a large droplet. When you have that decreased surface area because of the large droplet, where there's decreased access of the enzymes to the fat, you're going to have decreased chemical digestion. And so what the bile salts do is they break apart those lipid droplets into very small droplets by surrounding the outside of those. And notice they have a hydrophobic and a hydrophilic side, similar to the concept behind how your alveoli stay open in your lungs through surfactant. And so the bile injects right at the duodenum, right when the food's coming out of the stomach, and it emulsifies the fats. It breaks them down into very small droplets, and that allows for the chemical breakdown of that fat through the lipases. And this is looking at that emulsification in more detail. This is from the A&P textbook by McGraw-Hill. And it also gets into detail about how fats are actually chemically broken down. So I mentioned you have this big fat droplet or globule, as mentioned here, and the bile acids surround the outside and kind of break apart that fat globule so that it is smaller. So you've got these small little fat globules. Now the lipase, pancreatic or lingual, if it comes all the way from the mouth, can chemically break the bonds in the triglycerides to form what are shown here, monoglycerides and free fatty acids. Now fat digestion is very complicated. Those small fats get repackaged into something called a micelle. Basically, micelles have these small bile acids or small proteins surrounding the outside of the broken down single unit fats. So free fatty acids, cholesterols, monoglycerides, that kind of thing. This is also where fat-soluble vitamins get absorbed. So keep that in mind. Those micelles, we'll talk about it a bit, but when we get to absorption, we'll talk about that. Those are what are absorbed in the small intestine, and then there's extra stuff that goes on there that we'll talk about. Okay, so as I said, fat digestion is much more complicated than the other types of digestion. All right, so the gallbladder, I included this picture simply to show you the organization of the ducts and the gallbladder and the pancreas to the duodenum. So directly above where the duodenum's cut there, that's where the stomach is going to secrete, where you've got your the sphincter and the secretions, the chyme is coming out of the stomach. So it's a great location for the input of the bicarbonate and the enzymes and the bile to allow them to have their greatest effect. We have the bile coming from the liver through the hepatic ducts. If the person's currently eating, the bile will go straight down and through the papilla into the duodenum. If the person doesn't need the bile right then, it'll go into the gallbladder and it's stored in the gallbladder. And then CCK will feed back and cause the gallbladder to contract and constrict and expel the bile into the duodenum when there's a bolus of food. Notice that the bile duct meets up with the pancreatic duct at the duodenal papilla. And so you've got bile and pancreatic enzymes entering into the duodenum at the same place. There is an accessory pancreatic duct, and I would hypothesize that allows for sooner exposure to the bicarb to help with the neutralizing of the acid, in addition to exposure to the enzymes. All right, so now that we've done most of the breakdown, we're going to talk about absorption. This diagram is from one of the texts that I like to refer to a lot. I really like how it organizes pretty much everything that we're about to talk about into one solid diagram. And so I would recommend looking this over now and then going through the rest of the lecture and then coming back to it and looking it over again, because it shows the chemical digestion in the mouth, the stomach, the small intestine, and at the brush border of the small intestine. And then it shows how the single units, so the glucose or the free amino acids or the micelles, cross the cells themselves. And then in the case of glucose and amino acids, those go right into blood and they go to the liver and are processed. Versus the fat is broken down from the micelles into even smaller units called chylomicrons. Those are too big to go into the blood. So they go into lacteals, which are an extension of the lymph system. Those lacteals then meet up with the circulatory system in the same place that lymph does, empty into that, and then those chylomicrons wind up going to the liver to be further processed. So like I said, fat, much more complicated. So again, look over this diagram, and there's more to it than just this diagram, but I like how this diagram helps to bring a lot of the, especially absorption, into it. Okay, so when we're talking about chemical digestion, we're breaking down or hydrolyzing, or we're adding water. And this allows for the absorption of those nutrients. Now, as I've already mentioned, you have to break them into small enough units that they can cross cells through transport proteins or through gradients, concentration gradients, and then also be able to fit into capillaries, so be able to cross into capillaries or into the lymph, as is the example for fats. So when we're talking about carbohydrates, we're breaking those down into monosaccharides. When we're talking about proteins, we're breaking those down into amino acids, sometimes small peptides. And when we're talking about fats, we're talking about monoglycerides or fatty acids. Okay. So chemical digestion it occurs in two different places. It occurs in the lumen and in the membrane, and we call that membrane contact digestion. When we talk about contact digestion, we're specifically talking about what's occurring at what's called the brush border of enterocytes. So if you go back to that slide that I was saying is a good overview, the villi of the enterocytes, which are the absorptive cells of the small intestine, the villi extend up and they create what's called a brush border. Within that brush border, between the villi, we have enzymes there, and that is why we have this what's called contact digestion. In a cavity, like in the stomach or in the intestine, that would be luminal or cavity digestion, and that's going to be the enzymes that are secreted by other stuff, so the pancreatic enzymes and so forth. Versus the enzymes at the brush border, those are going to generally be created by those cells. We also do have some intracellular digestion. As it says, sometimes there's peptides that need to be broken down into single amino acids, and there's the reorganization of the fats. Okay, this is a good breakdown of digestive enzymes and generally where they're located. We've already talked about salivary glands. They have amylase and lipase. The stomach has pepsin. As I mentioned, the pancreas is secreting lots of different enzymes. Amylase, similar to salivary glands. The trypsin, chymotrypsin, those are for breaking down the proteins. And then the lipase and such is for breaking down the carbohydrates. For breaking down fats. Lipase, fats. The other ones listed here, the intestinal mucosa, those are those brush border enzymes, and some of them, so enterokinase is going to activate some of the other enzymes, so that acts kind of like HCl does. Sucrase, maltase, lactase, those are breaking down the saccharides into monosaccharides. And then the ones in blue there, those are breaking down two amino acids. Hopefully you've already noticed the trend in color. So blue is what's happening to proteins, yellow is what's happening to fats, and red is what's happening to carbohydrates. And pterokinase is its own thing, so that's why it's kind of highlighted in green. Okay, so when we talk about absorption, it's important to think about surface area, because in order to be able to absorb all these nutrients, you need to have them come into contact with the epithelium. And so to improve the chance of that happening, you want to move the food around, but you also want increased surface area. So in our small intestine, there are three major adaptations that create this huge surface area, as you can see here, twice as big as a tennis court generally. One of them is that the small intestine is very long. It's well folded and maintained in the abdominal cavity. When you look at a section of small intestine, you will see what are known as circular folds. These are gross, anatomically visible folds in the mucosa that, as this says, increases the surface area by threefold. Now, if you zoom in and histologically look at the extensions off of those circular folds, you'll see lines of epithelium called villi, and that increases the surface area by 10. And then off of the individual cells are something called microvilli. And again, those microvilli are what create the brush border of the cells. And those increase the surface area by 20-fold. So in the small intestine, we get this huge increase in surface area because of especially the villi and the microvilli. And we don't see these we don't see villi, I should say, in the large intestine, which is what's shown here. So this diagram is simply to represent a review of the different layers, the different tunics, but also to show that the small intestine has these villi, whereas the large intestine does not. The large intestine has crypts, which are extensions down into the tissue, but they don't have extensions up to increase surface area. And the intestinal crypts have a different function than the intestinal villi. All right, so this diagram shows the villi, one villi extending up, and each of the little squares is representing a cell. And so you can imagine on that each of those cells you're going to have microvilli extending off of them. So notice the intertwining of the arteries and the arterial capillaries and the venule and the capillaries, and then the lacteals, which are the lymph part of this. So these parts of if we were to talk about anatomy, the cells themselves that are lining the lumen, those are the lamina mucosa. And then the capillaries and the vein and the artery and the lacteal, that's all located in what's called the lamina propria. So that's all that connective tissue area directly below the epithelium. And so when there are nutrients outside, so where that A is, if there were nutrients, they would have to come into contact with the cells, cross the cells, and go into those capillaries. Then the capillaries would carry them away. If it was fat, it would go into the lacteal. So in the cross section, you can see how you've got this tight connection, intertwining of the capillaries with the epithelium to help allow for this absorption within the intestine. And then this is looking at microvilli. This is a TEM image, so that you can see the actual brush border. And you can actually see microvilli on light microscopy as well, but they wouldn't be as distinguishable. And this shows you some of the vesicles that would be probably pinocytic, so they're going to be taking something from the lumen and bringing it down and through the cell. And again, these increase surface area by about 20-fold. And then you find those enzymes there, which are the contact digestion enzymes that those are associated with breaking dipeptides into amino acids or disaccharides into monosaccharides. Okay. So as we're talking about enterocytes and the small intestine, it's important to mention that there is a big turnover there. The life cycle of these cells is quite short, and we have lots of turnover, and it's a huge surface area, so it's a lot of work, a lot of effort and energy that goes into the actual maintenance of the small intestine and the lining of the small intestine. That's why some animals, like a snake that doesn't have to eat, have evolved to not have to eat more than once a month, and they will actually regenerate and then degenerate their intestinal lining. And we do see coordination between, or sorry, not coordination, correlation between the life cycle of the enterocytes and disease. So if the cells become infected, then we tend to see disease. We've talked about endocrine cells, secretory. Goblet cells are the mucus-producing cells, so they're going to produce mucus. And then absorptive cells are enterocytes. The new cells are down in the crypts, and then they move their way up and out, and they are derived into the type of cell that they're going to be for their 3 to 6 day life cycle. And then they're shed into the lumen, and some of them are recycled, digested and recycled, and some are exchanged. So there are different ways that nutrients can be absorbed. 3 main ones are found in adults. The endocytosis, that's not really occurring in adults. That's mostly for babies that are getting immunoglobulins from their mom. So active transport, passive diffusion, and facilitated diffusion are, as you should know, the main mechanisms by which we move things across a cell, and we see them throughout the digestive tract. Now, we don't have time to talk about each of these individually in terms of each macromolecule and how it's moved across, but you should be aware of the fact that when a nutrient is absorbed, it does have to cross those cells, and so it has to use one of these mechanisms to do that. Okay, this is a nice, just brief overview of where stuff is absorbed. As I said, the duodenum is the main source of absorption, and that will frequently extend into the jejunum as well. Those are the first 2 parts of the small intestine. By the time we get to the ileum, we're absorbing stuff that hasn't been gotten yet, and then in the colon, we get colon electrolytes. And I would also add to the colon anything that's broken down by symbiotic bacteria that might not have been available to be absorbed in the duodenum. Now, notice the stomach absorbs ethanol, and so that's why if you have an empty stomach and you drink alcohol, you can feel the effects fast because it is quickly absorbed in the stomach. So we're briefly going to go through the digestion, and this goes back to that diagram at the beginning. So in the mouth, you have amylase secreted in the saliva, and you do get some digestion in the mouth. Now, something that you probably haven't talked about previously, you can have up to 40% of your starch digestion occurring in the stomach. Now, you might say amylase is going to be broken down by the acidity in the stomach, and that's true, but we're forming kind of this bolus of food in the stomach that's becoming chyme, that's being mixed. At the center of that part of the digested material is not coming into contact with the HCl, and so that amylase stays intact. So at the center of kind of the bolus that's in the stomach, that is still undergoing amylase digestion. Further digestion occurs in the pancreas with the amylase there, and then we have contact digestion occurring at the brush border to go from disaccharide to monosaccharide, so from sucrose to glucose. Okay. And this is a nice overview from your book. Again, this is very similar to what's shown in that diagram I showed earlier. So you've got amylase breaking down into what's called maltose. You might also eat food that has lactose or sucrose in it. That gets broken down at the brush border, contact digestion into glucose or fructose and galactose as well. And then those monosaccharides are what cross the cells, get absorbed into the blood, and then further processing occurs in the body. All right, so let's look at proteins. We want to go from proteins to peptides to amino acids. This doesn't start until the stomach. There is this doesn't start until the stomach. And specifically in the lumen of the stomach, we have pepsin. We've already talked about that. That's going to start the breakdown of the proteins into peptides. We get into the small intestine. We've got the three different major proteases: trypsin, chymotrypsin, and carboxypeptidase. I'm not going to go through the specifics of how what bonds all of these break and how all of these work. But you should recognize that in the stomach, you're going to go to smaller peptides, and then in the small intestine, you're going to go down to dipeptides, and then at the brush border, you're going to generally break down into amino acids. If we don't get to amino acids in the brush border, then in the mucosal cell itself, we'll get broken down into amino acids through the peptidases. So that's what this is showing again, and amino acids wind up being absorbed into the blood. All right, so I've already mentioned fat is the most complicated, and it's assimilation because we're not just breaking it down, we're also reforming things. So we go from triglyceride to monoglyceride and back to triglyceride. And that's because in order to be absorbed into the cells, we have to have this conversion. So in the duodenum, in the lumen, we break down with lipases into one fatty acid into monoglycerides. And then within the cell, within the enterocyte, we reform that into triglycerides. And I included the diagram from earlier here to help. So most of what you eat is going to be triglycerides, and that's going to be broken down through emulsification, which is a physical breakdown. That's not chemical, it's mechanical. That allows the enzymatic digestion to occur, so that allows the lipases to have access to the triglycerides and break them into monoglycerides and fatty acids. The monoglycerides and fatty acids form into micelles, and that is a consequence of the bile acids and how they are transported because they are hydrophobic. Micelles cross into the cell, into the enterocyte. So there's no contact digestion here. Micelles cross into the enterocyte and then get taken apart and reformed, so assimilated into what are called chylomicrons. And those chylomicrons, which are kind of this protein shell containing triglycerides, that's what gets absorbed into the lacteal. All right, and so this is showing that with a little bit more detail. So we get micelles that get absorbed, and we also do the free fatty acids, and the monoglycerides can also cross. The enterocyte itself has forms triglycerides and apoprotein. That is that what's shown before was that protein shell, that like green shell surrounding the triglycerides, that forms the chylomicrons, and then that is what is absorbed into the lymph vessel. All right. Go back and look at that diagram again from earlier and review the enzymes and be familiar with them. And then bring this all into perspective with what we talked about in the first section about how this is being moved through and the feedback from hormones and all of that. Oh, I included these lists just to help.